Welcome to another Chapter 6 virtual lecture on microbial growth. We spent a previous virtual lecture talking about concepts like binary fission, the phases of bacterial growth, of which there were four, if you recall. There were lag phase, log phase, stationary phase, and death phase. Now I want to talk about what are some of the requirements that assist in that growth. And what are some of the conditions in which growth may be stunted? Meaning, what are some of the conditions that, if met, cause bacteria to enter the death phase? Talk about chemical requirement, nutrient requirement. So here's a nice introductory slide. Organisms use a variety of nutrients for their energy, and they need to build their organic molecules and cellular structures. An organic molecule is a carbon-based molecule. And there are four major classes of carbon-based molecules that are very important in producing these cellular structures. The first class would be carbohydrates. The second class would be lipids. The third class would be proteins. And the fourth class would be nucleic acids. Examples of which include DNA and RNA. Now, we've seen this already if you think about it. Review the prokaryotic cell structure. Was there any part of that cell that was rich in carbohydrates? One example would be the cell wall. If you recall, the glycan part of peptidoglycan had those repeating NAG and NAM subunits, which form that disaccharide. So we know carbohydrates are very important in allowing for the function of that cell wall. Can you think of another cellular structure that's rich in lipids? That's right, the cell membrane had many phospholipids, and those phospholipids were very important in allowing the cell membrane to be selective. How about proteins? Can you think of cellular structures that are high in protein? Well, certainly the cell wall consists of protein. If you recall, the cell membrane has proteins in it. The peptido part of peptidoglycan consisted of amino acids linked together to form cross bridges that were linked together to provide stability to the cell wall, which allowed it to resist overexpansion of the cell. Cell membranes, both the outer and the inner cell membrane, were rich in proteins. The outer membrane of a gram-negative had those porins. We know the inner membrane has transport proteins, both of which aid in getting things into and out of the cell. So listen, folks, in order for me to make the cell wall, if I'm a bacteria, in order for me to construct the cell membrane, I need to build these organic molecules, and I need energy to put it all together. So there are strategies that bacteria use to get that energy. There are strategies bacteria use to build those organic molecules. That's what we're going to talk about over the next few slides. So what are some of the common nutrients that cells use for energy and use to build their organic molecules? Well, one of the things we have to understand is these nutrients have to have a source of carbon, have to have a source of nitrogen and hydrogen. We also know oxygen can be important for cellular growth. So the question becomes, where do we get these nutrients from? And the answer is not as simple as it would appear. Depending on the bacteria, they can get those nutrients from a variety of sources. So let's start here with some basic chemistry. I've listed on the previous slide nitrogen, carbon, oxygen. I've even included some others you may be familiar with, like phosphorus and sodium. If I were to ask you which element out of those five is the basic building block of all biological molecules, could you tell me based on what we just learned? Again, press pause, go to poll4.com, put in your answer, or if you want to, send a text with your answer choice, like 30902 to 37607. All right, based on what we just talked about, carbon is the building block of all molecules. All those molecules like proteins and lipids and carbohydrates are all carbon based. So the correct answer here would be carbon. So where would bacteria get their carbon from in order to build these organic molecules? And to extend this, we're going to see that carbon-based molecules are also a good source of energy. So where can bacteria get the energy from in order to build their cellular structures? Now here's a nice figure. It's not in your textbook. I took this from someplace else, but I think it's good to study from. There are 4 basic groups of organisms, and we group them based on 2 things. Where do they get their energy, and where do they get their carbon? Let's first start with the energy source. All living things need energy. Whether you're a human, a bacteria, and everything in between, all cells need energy to carry out cellular work. We saw a great example of this in Chapter 4. Without energy, cells cannot carry out active transport. Without energy, cells can't build their cell wall. Cells can't make their cellular structures without energy. So where can bacteria get their energy from? There are 2 common sources. First would be from light. Second would be from chemical compounds. Well, let's tackle this first one. Light. What's the ultimate source of light on the planet, and therefore the source of energy for bacteria in this class? That's right, the sun. The sun is a great source of light energy. Therefore, we call bacteria that fall in this group phototrophs. Photo meaning light. On the flip side, alternatively, those bacteria that cannot use the sun and therefore require chemicals or rely on chemicals to get their energy are called chemotrophs. I'm going to tell you, as far as an allied health major is concerned, the pathogens, i.e. the bacteria that make us sick, are chemotrophs. And the reason they want to inhabit our body is because we're a great source of chemical compounds. Let's see if this makes sense. We have bacteria that if you ingest in certain amounts can cause food poisoning. Food poisoning is a condition in which bacteria are residing in your gut and they're producing toxins that cause inflammation. Now, would you say there's a lot of sunshine that gets to your colon? Of course, the answer is no. I know there's an analogy about sun shining up a certain part of the body, but in our case, that's a dark place. So bacteria that inhabit your colon cannot rely on light for energy because the colon does not allow light in. However, in your gut, there's certainly lots of chemicals, either from the food you just ate or from the cells that make up your colon. So most pathogens are going to be chemotrophs. So now let's move on to the carbon source. For some bacteria, using a simple molecule that's readily available in the environment, carbon dioxide, they could take that carbon dioxide and build all their important cellular structures. We would call these organisms autotrophs. Auto meaning feed self. Just basically using the air that's supplied, these bacteria can harvest the carbon that's part of carbon dioxide and build all their proteins, all their carbohydrates, all their lipids, all their cellular structures. However, most bacteria can't do that. They cannot harness carbon dioxide to build their cellular structures. So what they rely on are organic compounds from their environment. Now, I listed the 4 major classes of carbon compounds 2 slides ago. In the window that's going to pop up, can you list the 4 classes of carbon-based molecules? That's right. If you list through, the four major classes include carbohydrates, lipids, proteins, and nucleic acids. So what that means then for heterotrophs is they need to obtain carbs from their environment, proteins from their environment, etc. That's what a heterotroph does. Well, that makes milk a great source for organic compounds for a bacteria. That's why milk can spoil, because the bacteria can break down the milk protein in there, which causes the milk to sour. So now when you put this all together, you have photoautotrophs, which use light and carbon dioxide in order to get their energy and to get their carbon. Plants fall into this group. Algae fall into this group. As far as the prokaryotes are concerned, cyanobacteria fall into this group. These are photosynthetic organisms. I think you've heard of that before, right? Plants use the sunlight to feed themselves. That's photosynthesis. There's also a slightly different class of bacteria called green sulfur and purple sulfur bacteria that don't produce oxygen, that don't use water. They're slightly different. Now, on an exam, I'm not going to ask you about green sulfur and purple sulfur bacteria. What I do expect you to know is there are bacteria that can use light for energy and use carbon dioxide to feed themselves. There's also chemoautotrophs. Chemoautotrophs require chemicals for energy. However, they can use carbon dioxide to build organic molecules. These are hydrogen, sulfur, and nitrifying bacteria. Again, I'm not going to ask you to list those on an exam, but we should know there are some small classes of bacteria that fall into this group. The group that we're going to focus on most this semester is this last group. I would expect you, as we go on in this semester, when we get, for example, when we get to chapter 11 and chapter 12, you should be able to describe for me what a chemoheterotroph is. A chemoheterotroph uses organic compounds both as an energy source and as a source of carbon. A great example of that is you. You need to eat a diet or consume a diet rich in carbohydrates, rich in lipids, rich in protein. So not only can you get the carbon for your cells, so you can get the energy for the cells to use that carbon. So most animals fall into this group. Fungi, which we'll focus on in chapter 12, protozoans, which we'll focus on in chapter 12, are chemoheterotrophs. Many bacteria are chemoheterotrophs that all require oxygen, which is a concept we'll touch on in the next few slides. They're aerobic in nature. There are also bacteria that are chemoheterotrophs that cannot grow in oxygen. So they prefer to grow without oxygen. We call those anaerobes. There are some animals that can carry out anaerobic respirations, some protozoa, which again will fall into chapter 12, and then some bacteria, which will be the focus in chapter 11. Lastly, some bacteria can grow with or without oxygen. Those are fermentation or fermentive bacteria, and even yeast, which again we'll learn about in chapter 12. Those are chemoheterotrophs that when they use the chemical compounds for energy, they can use a special pathway called fermentation. Most, if not all, pathogens, which again what we tend to focus on, fall into this category of chemoheterotrophs. So what do we need to know for an exam? If I were to ask you to define a phototroph or a chemotroph, you should be able to do that. If I ask you to define an autotroph or a heterotroph, you should do that as well. Also, I want you to have an appreciation that bacteria can fall into any one of those four groups. So what we can see here, our bacteria are quite diverse in where they get their energy from and where they get their carbon from. But as far as the ones we study in this class, because we tend to focus on disease-causing bacteria, most are going to be chemoheterotrophs. So now that we've got the source of carbon out of the way and the source of energy, let's move on to oxygen. We began to touch on that previously. If an organism requires oxygen, what did we call that based on the previous slide? Now in the box that pops up, make sure you focus on spelling here. That's right, aerobic organisms require oxygen. Conversely, if you don't require oxygen, what did we call those bacteria? Again, spelling will count here, so be careful. That's right, anaerobic bacteria. So we're going to see, even amongst this group called aerobic and anaerobic, there are degrees of which oxygen is required. I think this is very important as you go forward. We often classify bacteria based on this property, as we just saw. So as we discuss the next few slides, make sure you learn the terminology, like strict aerobe, strict anaerobe, and facultative anaerobe. So for those bacteria which can only grow when oxygen is available, we call those obligate aerobes, or another way to say that is strict aerobes. What that means is they have a strict requirement for oxygen, and if oxygen is not available, these bacteria ultimately enter death phase. The numbers decrease rapidly. Actually, if you think about this, humans are also obligate aerobes because humans require oxygen. Don't do this experiment at home, but if you go without oxygen long enough as a human, your brain shuts off first. You go brain dead. Why? Because without oxygen, you can't make energy. The brain cells starve to death. And if you continue on, your heart cells stop because your heart needs energy, and without oxygen, you can't harvest energy as well. We also have this group called obligate anaerobes, in which oxygen is toxic. Now this goes against everything we've ever learned. I always think about oxygen being a good thing, because if I go without it, I die. But for some bacteria, oxygen is toxic. And even for humans, oxygen is toxic. You may have heard of this before, antioxidants. Antioxidants are molecules or chemicals that you find in a variety of food products, and you even find in supplements. And they're supposed to protect us against oxidants. So let me define what an oxidant is. An oxidant is an oxygen, let me put it this way, a form of oxygen that damages cells. And in humans, that damage to your cells can lead to a variety of things like cancer, can lead to stroke, can lead to a heart attack. Essentially, damage to the cell is part of what contributes to the aging process. So in that sense, oxygen is toxic even for our cells. However, we've evolved ways to try to manage that. Now, does anyone know any food products, just for the heck of it? I'm not digressing here. I'm not talking about bacteria for a moment. But does anyone know any food products that are naturally high in antioxidants? That's right. You may have heard of red wine being high in antioxidants due to the fact that the skins of grapes are high in antioxidants. Vegetables can often be high in antioxidants as well. And of course, you could take supplements. So the role of those antioxidants are to protect our cells from these toxic oxygen species. Well, it turns out some bacteria that are obligate anaerobes don't have that protection. And so what ultimately happens in obligate anaerobes is as oxygen becomes available, those obligate anaerobes die. So what are these toxic forms of oxygens? I'm just going to list them here. I don't want you to get too deep into this, but the first toxic form is superoxide, which essentially is O2 negative. So it's an oxygen with an extra electron. That gets formed during energy production. So it's actually a natural product We naturally produce superoxide. So for aerobes, they have enzymes called superoxide dismutases whose job is to detoxify them. Superoxide dismutase is an antioxidant. That's why aerobes can survive in the presence of oxygen. However, anaerobes lack that enzyme. Therefore, these superoxides radicals, these superoxide species build up, and what they do is they damage the cell, and if that damage is so extensive, that cell ultimately dies. That's why an anaerobe dies in the presence of oxygen. Another toxic form of oxygen is peroxide. You may know one form of this that's common, which is hydrogen peroxide. Again, this is a natural byproduct of energy production. Aerobes handle hydrogen peroxide by producing enzymes called catalase and peroxidase to detoxify it. I'll show you that on the next slide. However, obligate anaerobes lack those enzymes or don't produce enough, so as hydrogen peroxide builds up in the cell, so does damage, and ultimately, if the damage is so extensive, the cell dies. Here's why that's helpful to me. Let me give you a big picture idea here. Let's say someone has a disease called gangrene. Gangrene can often be caused by an anaerobe, and the reason being is they don't have good circulation to their extremities. You may recall that foot that we saw in chapter 4 that was black and rotting, showing necrosis of that tissue. Well, what happens in those individuals is due to poor blood circulation, the amount of that obligate anaerobe begins to go up, and as it goes up, it begins to produce toxins that destroy the tissue. So one way we can treat early signs of gangrene is using a hyperbaric chamber. You may have heard of a hyperbaric chamber before. That pumps oxygen into that tissue. And because the bacteria lacks the enzymes to get rid of the toxic forms of oxygen, ultimately we can clear that infection. So one way to treat infections due to anaerobes is to use oxygen like medicine. Oxygen is going to kill those bacteria. Here's sort of a summary of what I was talking about here. Again, you don't need to learn the chemical reactions, but what I want you to understand is aerobes produce superoxide dismutase, produce catalase, produce peroxidase. All three of those are enzymes that detoxify oxidants. We could actually use this as a test. We can classify bacteria based on their ability to produce catalase. What you would do is you would take some bacterial cells, so you would add cells to each slide. So I have bacteria A on this slide, bacteria B on this slide. And then what I would do is I would drop on hydrogen peroxide. Now, for cells that don't produce catalase, there is no reaction. Nothing occurs. But for cells that produce catalase, what happens is they convert hydrogen peroxide to water and oxygen gas. And because that gas is produced, can everybody see that? We have those characteristic bubbles that arise. Actually, you can do this. You know, humans are aerobes, correct? So would you hypothesize that humans produce this enzyme, or would you say humans lack this enzyme? That's right, humans produce catalase. And so an interesting experiment is if you have a wound and you try to add hydrogen peroxide to clean it out, you'll actually hear the wound begin to sizzle, or you may see it bubble. And the reason being is your cells produce the catalase, so as you add hydrogen peroxide, O2 gas is released, causing your wound to produce those bubbles. That's why hydrogen peroxide is not very effective if you want to clean out a wound, because essentially what happens is you neutralize the hydrogen peroxide. You break it down. And that's why you see that bubbling. So I think that's extremely important concept. Not all bacteria like oxygen. We're not used to that, right? We're human-centered. We know without oxygen, humans die. Now I'm introducing a concept which states this: not all living things need oxygen. For some living entities, oxygen is damaging. Oxygen is poisonous. So here, I'm introducing a new group. I've already talked about aerobes. They need oxygen for energy. And that's a concept we're going to introduce next chapter, chapter 5. Anaerobes do not need oxygen. They do not use aerobic metabolism. Oxygen is toxic for anaerobes. Now, this third group, facultative anaerobes, that's an interesting group. See, for facultative anaerobes, when they have oxygen, they're okay. They use that oxygen for energy. However, if you take their oxygen away, they can continue to grow using alternative energy-producing pathways like fermentation or anaerobic respiration. Again, this is probably going to be the main concept I'm going to discuss in the next chapter, chapter 5. Right now, here's what I need you to know. Facultative anaerobes grow with oxygen or they grow without oxygen. Facultative is a term we're going to use throughout the semester that means with or without. So if you were to take a test tube here, and this represents solid media, sort of like gelatin. And because it's solid, here's what I'm going to tell you. As oxygen tries to diffuse in, oxygen can't get in very far because it's solid. So as you get towards the bottom, there's really not a lot of oxygen present. So notice here, for an obligate aerobe, the bacteria are all growing on top where oxygen is plentiful. Here in an obligate anaerobe, you notice the bacteria all growing on the bottom where oxygen is absent. Well, notice now the difference highlighted here by a facultative anaerobe. For a facultative anaerobe, you have growth at the top, you have growth at the bottom, you have growth in between. They grow in all zones of the tube because if they have oxygen around, that's great. They grow very well. If they don't have oxygen around, that's fine. They continue to grow. So they're able to flip the switch. Now let me give you an example of a group of bacteria that are facultative anaerobes. The bacteria that reside in your colon. See, if you were to describe your colon, if you recall, your colon is highly folded, right? Your intestines are actually quite long, and they're folded up so they can fit in your body. And that increases the surface area so you can absorb nutrients. But that's a concept that's meant for A and P. For us, here's what's important. Due to the winding nature of the colon, the oxygen levels vary throughout your colon. Sometimes there's high oxygen levels, sometimes there's low oxygen levels. So how can a bacteria live in your colon, for example, E. coli? And the answer is simply this. E. coli is a facultative anaerobe. When oxygen is available, it'll use it, it'll grow. When oxygen is absent, it'll still grow despite that fact. So it can grow under a variety of oxygen conditions. Here's actual test tubes. It may be difficult to see, but you can see how cloudy it is here at the top for an obligate aerobe. Notice for an anaerobe, there's clear right here at the top because of the presence of oxygen. And notice facultative anaerobes, there's no clear spots. Growth is everywhere. We call that an aerotolerance test. All right, let's see if you get this concept. Now, here's a question that is going to be familiar to you once we get done with this chapter. I'm going to show you only the log phase growth. So there's no lag phase here. There's no stationary phase. There's no death phase. I'm just showing you that part of the growth curve which represents maximal growth, which represents the population doubling. And here on the y-axis I have log number of cells. Here on the axis I have time. Recall that how steep the slope is directly impacted by how well it's growing. If cells are growing very well, we're going to have a very steep slope. If cells are not growing very well, we're going to see it be flatlined. So now here I'd like you to vote. If I have an obligate anaerobe, which line represents its growth in the presence of oxygen? A, B, or C? Press pause now and log in your vote. That's right, the best answer here is C. Now the reason is this. If I can go back. Anaerobes do not use oxygen. In fact, obligate anaerobes lack the enzymes to grow, so oxygen is toxic. The oxygen that builds up damages the cell and results in cell death. Well, there's no better picture of cell death than C. The number of cells is not increasing over time. So you can see why this would be a good treatment for obligate anaerobes causing an infection. If you give them oxygen, they begin to die off. So again, if you have questions on that, please let me know. I think it's fairly straightforward. Some bacteria like oxygen, some bacteria hate oxygen, and some can go either way.